In this video, we will be talking about sonic hedgehog signaling and the relation to neural tube formation in vertebrates. Some background. It gets its name from the polypeptide ligand, an intracellular signaling molecule called Hedgehog, which is found in a study on Drosophila of fruit flies. Those lacking the Hedgehog gene look like an embryo with pointy tentacles which resemble a hedgehog, thus the name. It is essential for many aspects of normal embryonic development, like the formation of the neural tube, because it transmits information to embryonic cells required for proper cell differentiation. Hedgehog proteins help facilitate the activation of the signaling pathway, and there are three of them: desert hedgehog, Indian hedgehog, and sonic hedgehog. The activation of the signaling pathway is essential since the roles of signaling include but are not limited to facial morphogenesis, forming midline of the body, neural differentiation, and tissue growth. One important concept to note in regard to sonic hedgehog signaling is graded signaling. This is essentially the signaling which controls the expression of transcription factors which pattern the dorsal-ventral axis of the neural tube. The hedgehogs are characterized as morphogens, which have the characteristics of being able to induce different responses at different thresholds. Hedgehog signaling in vertebrates occurs within the primary cilium. Primary cilia are located on the surface of the cell and gather information about the environment. They are also attached to microtubules and a protein called GPR161. Within the primary cilium, there's an inhibitory receptor called patched. Smoothened is a signaling receptor inside the cell, which is usually within an endosome. When there is no hedgehog protein present, this means the pathway is inactive. And when it is inactive, patch will inhibit smoothened, and when inhibited, it cannot enter into the plasma membrane, so it stays in the cytosol bound to the endosome. Whilst in the inactive pathway and smoothened being inhibited, Gli proteins like Gli2 and Gli3 are associated with suppressor of fused proteins or SUFU proteins. When inactivated, proteins like CKI, protein kinase A, and glycogen synthase kinase 3-beta or GSK3-beta, they will all phosphorylate Gli proteins. When phosphorylated, they are cleaved by the proteasome into Gli2R and Gli3R, which are the repressor forms of these proteins. These repressor proteins can translocate into the nucleus where they can inhibit transcriptional activation of hedgehog-related genes. Now, let's talk about when hedgehog proteins are present. The signaling pathway is now active. So when sonic hedgehog is present, it can bind and inhibit patch, which makes patch no longer able to inhibit smoothened. Once the sonic hedgehog protein is bound to patch, both GPR161 and patched leave the membrane and get degraded within a proteasome in the cell. Smoothened is no longer inhibited by patch and gets phosphorylated by CKI and GPRK2. The now phosphorylated smoothened can enter into the plasma membrane in close association with a protein called EVC. KIF7 aids in the movement of both Gli proteins and SUFU proteins through the primary cilium, and the Gli proteins dissociate with the SUFU proteins, making them no longer bound to each other. These Gli proteins have not yet been phosphorylated, which means they are not processed by the proteasome, so they can remain in their full form and not the repressor form. So Gli2 and Gli3 can translocate and enter the nucleus and bind to the DNA to induce expression of Gli target genes. Gli target genes are responsible for development patterning and differentiation. Different morphogen gradients along the dorsal, ventral, and anterior posterior axes influence the development of the neural tube into the brain and spinal cord. Sonic hedgehog signaling is essential in the formation of the dorsal ventral axis of the neural tube and is first produced in the notochord. This causes the medial hinge point cells of the neural folds to become the floor plate of the neural tube. Floor plate cells secrete sonic hedgehog, which creates a concentration gradient where the highest concentration of sonic hedgehog are at the floor plate. As it is being secreted, the concentration decreases as distance increases from the floor plate where it was initially being produced. A cell can identify its position as being in the ventral nerve depending on the amount of sonic hedgehog produced. Based on the amount of sonic hedgehog, the cells divide into a number of subpopulations like inner neurons like V3, V2, V1, or V0, or even motor neurons. The hedgehog pathway has signals for developmental patterning of the brain and face, and interference yields birth defects like cleft lip or cleft palate. The hedgehog signaling pathway is essential for many aspects of normal embryonic development, including formation and patterning of the neural tube. Absence of the sonic hedgehog ligand is associated with the midline defect holoprosencephaly, when the brain fails to divide into two hemispheres during early development. This can also affect development of the head and face.